please be careful. And I think I can skip who am I? I think the slide about me, but let me start from here. The product I was involved when I work in the startup companies. So when I built Nanointech, and I worked 5 years in there, and I developed several microfluidic devices, and some are cell counter, and some is the neon transfection system, and the other one is the microfluidic replicate. And I also developed a cell counter. This was a cell counter and cell culture platform. And among this product, this cell counter devices and one replicate are strongly involved with the nanointerstice technology to enhance the fluid filling into the very tiny region of microfluidic channel. And this company, Nanointech, is successfully going to the coastal market in 2006. And now currently we have a market capital about $200 million. And last year it was acquired by SK Telecom. And also another spin-off company from the Nanointech is Lowe's Biosystem, and it just passed the coastal evaluation and will go into the coastal entry next year. And also this company has the annual sales about $4 million and with 9 employees. And also I made a company in Singapore. The name is Biotech, and it involved with the 3D cell culture platform. And this is the C-chip disposable cell counter, so that is the patent. And this is simple microfluidic channel with 100 micro channel height, and with the length is about 10 centimeter, and the liquid inside of the channel is about 20 microliters. And this is distributors, and we have annual sales about $4 million. And the unit price is about $1 per chip. So that means about 4 million chips was sold worldwide in a year. So this is microfluidic chip made of acrylic plastic. And this is the usual cell counting procedure before applying our product. So this is glass hemocytometer with a laser grid, and the people fill the staining solution of the cell into the devices, and then measure under the microscope. And the serious problem, it has the grid line. So serious problem with this device is they're very difficult to use because they have to assemble it, and after use they have to disassemble it and to clean it by themselves, and very expensive because the grid can only fabricated by laser ablation, and also but very easily breakable. So we adapted the mass production line of the CD and DVD, and this is the mass production line of the DVD, not ours. So usually the CD DVD line has to fabricate the nickel stamper. It has the nano or micro patterns made of nickel, and it should be installed into the mold. It should be installed with the mold. So it has the as you are very familiar with the shape, it's very tiny pattern of the CD, and it is the nano pattern features patterned on the nickel plate, and this nickel plate is integrated with the injection mold system, so the people can easily fabricate, mass produce the CD and the DVD with a very low price. And this is the factory, one of the factory of our company, and this is the nickel stamper with a very small, tiny grid on it, and this is the installed the mold to define the external structure of the chip. And this is the total mold because I'm a mechanical engineer. So this is an injection molding machine. So you can see usually it was used to fabricate the DVD or CD, but you can see this is modified to fabricate the tiny chip, microfluidic chip substrate. So this is the fabricated substrate. So it has a 1 inch width, and then 3 inch height, 3 inch length, and it has the microfluidic or nanopatterns or any other things. And only product this is pretty cheap because this is mass producible and injection moldable. So we can fabricate the substrate, one substrate of the microfluidic devices within several cents. So, but at first time we have to codify our hand and then wash it with the shower and then to do the surface treatment and then assemble by hand. But and finally we made a really nice factory, but I will show you later. And this kind of microfluidic chip can be applied to the automatic cell counter because the people can detect or count the cells by their hand, but also we have a very simple camera system. This is very simple camera, and we can put the chips into the devices, and then they take a picture, and then just count the cells by the software. And this is annual sales, and this is one of the impressive release of the Life Technology in 2009. The Countess was one of the company's most successful product launches in history. And this is how to use the microfluidic chamber for the cell counting. So this is cell culture solution, and this is completely disposable and very easy to use, and very tiny with a very tiny spacing and very precise channel height. And user can just put the chip into the machine, and then they take pictures. And when you push the button, they automatically count the number of the cells. And this is one of the advertisement from the Life Technology. So.
Oh, look at the one color to play. No, I need to count these stupid cells. I've lost count again. This is taking too long. Counting's so boring, and I'm just bored of standing here.
So, in this case, he's very smart, and that's why he suggests our product to his hired friend.
Try this, the Countess. It does all the counting and dilution calculation for you. It's so easy to count cells.
Yeah.
Cool.
With the microfluidic technology. And one problem is the reason why they run out of the out of the lab after getting such a nice product, by the way. And this is the Interstellar movie, and you can see the Countess is also installed in the future. So the future laboratory. So this is a Countess. So the Countess has been very successful, and we have plenty similar product worldwide. So they also have the microfluidic channel devices and developed a very similar counting systems. And this is another system made by Nexalog, the German company, and they have also laminated microfluidic chambers. And also Bio-Rad also developed a very similar platforms. But you can see the microfluidic chamber is a pretty short and shorter than the normal microfluidic devices. And this is also our new lens-free portable cell counter. And if we use the adapted lens-free system like that, so we can, if we do not have lens in our devices, take the picture taken, the cell picture taken, just look like that. And this is the picture taken under the microscope. So we can reconstruct this kind of more images into the cell images, so we can count the cells even without the lens. So that means we do not have to install the very expensive lenses, and also we dramatically reduce the size and dimension of the devices, and also it's very easily adaptable. And even for the humidity chambers, it's directly used for the humidity chamber because there is no chances for the capturing the moistures between the lens. So this is a new product. And also another thing is that if we can increase the channel length, we can take the multiple pictures, and it will dramatically enhance the precision of the measurement. So that is FDA certified leukocyte counter. And one important thing is that we use the exactly similar devices, and we developed a very similar detector system. But if the system or protocol is FDA certified, we can dramatically increase the price of the chip. So that means that we can acquire the 92% profit after selling the chips. So this is the microfluidic channel for the FDA certification. You can see the length is more than the 40 centimeters. But there is a serious problem because that is made of plastics, and plastics are generally hydrophobic. So after surface treatment, after surface treatment can make the channel surface hydrophilic, but after 1 year or 2 years, it dramatically restores original hydrophobicity, so it is impossible to fill the liquid into tiny microfluidic chambers. So it has 100 microchannel height, but this is moving. So you can see when I in such a long time, it cannot fill into the liquid, and that is a serious problem in the mass production or commercialization of the microfluidic devices. And this is the reason. So usually in experiment or not in the company and in the development status, we can use the chips after surface treatment. So you can see the 30 degree or 40 degree and 50 degree of the contact angle, and that is completely hydrophilic after oxygen product treatment. But after 3 months, the surface restores original hydrophobicity, and after that, we do not have any more chance to fill robust filling into the microfluidic channel. So we start to install the nanointerstice at the side wall of the microfluidic devices. And so this is the nanointerstice, and channel height is very tiny channel with the 1 millimeter spacing and only 100 or 200 and even up to the 800 nanometer channel height. So you can see the channel is filled first into the nanointerstice because it has very strong, very low meniscus, very small meniscus. That means very high pressure. So it can dramatically enhance the liquid filling into the normal microfluidic channel. And as you can see, because the high pressure between the very small meniscus of the liquid in the nanointerstice dramatically affect the larger pressure to the liquid filling of the main channel and dramatically enhance the liquid filling of the microfluidic channel even without any surface treatment. So this is the equation related to, and so we can precisely estimate, we can precisely calculate the liquid filling velocity or any other things. And as you can see, after nanointerstice installation, you can see the dramatically enhance the liquid filling into the microfluidic channel, and you can see very small amount of the liquid fill first into the nanointerstice, and it directs the fluid in the main channels. And when we the channel height is only 100 micrometers, but when we dramatically reduce the channel height like 20 micrometers, and even in this case, you can see the liquid filled into the nanointerstice at the side wall of the channel, direct the main fluid and help the liquid filling into the microfluidic devices without any surface treatment. So this is another important advantage is that is very easy to fabricate, and also we could acquire final tolerance of about 2 micrometers in the 1 million chips per month mass production line, and that is pretty important because the 2 micrometer tolerance means that 2% errors. So we have to reduce it. We have to minimize it as much as possible. Then we can make the tolerance within. We can control the tolerance within the 2 microns in the mass production line. And so this is first production line, and this is second turret bonding machine line. And you can see the turret bonding machine, the movie of the turret bonding machines to increase the production of the line. And this is the another automated line, and this is final production line of the microfluidic devices. So you can see the the fully automated and make the 10 chips at the same time to dramatically increase the production rate. And I think I can, okay, I have about 10 minutes left. Okay, so this is another one, neon transfection system, and use pipe tip as a microfluidic chamber. And this is the various method of the transfection. So this is, we can, if we want to transfect the cells, we can use a lipid or polymers or virus and other things. And electroporation is one of very nice method to directly punch or make the hole at the outer lipid bilayer of the cells, and then put the DNA into the cells. So this is electroporation method, and previous devices, this is made up by BTX, and electroporated by BTX, and then they sold by Harvard operators, use the cuvette to fill the liquid with the DNA solution and install the electrode at the side wall of the cuvette. So that means this amount is pretty high, and another problem is that it has a very wide the width of the electrode, and that means it made a lot of defect or contamination and etch effect and bubbles, and that's why the success rate of the electroporation is dramatically lower. And we can use the microfluidic chamber, but there is no way to collect the liquid in the microfluidic chamber like that. So we made the idea to directly use the pipette tip as the microfluidic chamber. So this is we use the pipette tip. So first process is just to fill the liquid into the pipette tip, the cell and the DNA solution into the pipette tip, and it has the electrode at the top. And when you put the pipette tip into the machine, it has the electrode at the bottom. So this machine generate the electric shock in the cells in the pipette tip, and we can directly use the pipette tip as the microfluidic chamber, and it is pretty easy to bring it back to the petri dish. So this is the very important product of the Live technology, and we sold 2 patents for the $30 million. And okay, I think I can skip it. So currently, I'm now mainly working for the precision medicine applications, because recently after the president's speech, everybody thinks that the precision medicine is very important. And one of the most important reasons why the precision medicine is important, especially in the oncology, is that the response rate of patients for the oncology drug is only 25%. That means only 25% of the patients, the oncology drug will work. So it's a really important problem, a serious problem, and that's why we need the precision medicine, especially for the oncology applications. So we have developed several methods. My lab have developed several methods. So for example, we can analyze the cells or some particles or antibodies related to the cancer with the blood test. And also we can collect the CTCs in the microfluidic platform or other platforms. And we also developed the CTC collection and the culturing, 3D culturing system. And also we directly acquire the tissue biopsy of the cancer and then do the cell screening. And that is called the PABTS system. And now my company in Singapore is doing commercialization of this whole process and services. And for the blood test, we need the quantity of microfluid in the assay, because the PSA or other cancer-related antibodies or hormones should be quantified in the liquid. We do not need the information about the on-off or yes or no. This is not important at all. We need the precise information about how much one molecules or antibodies in the human blood. So this is we exactly use the similar devices for the diagnosis, and then you can see that with the Nanointerstice, the liquid is easily filled into the chamber, but we have a very nice control system to make the flow to filling stop and go and stop and go, and we can precisely control the individual steps of the liquid filling and the analysis. So this is why we started a new company, Absol, for the development of a microfluidic contactile replicate. But I'm sure I could not go into more details only because this is precisely the steps in the Absol did not want point. So I think I have 5 minutes left, so let me go into briefly introduce some recent work about the cell screening. So recently, the precision medicine is mainly on the genetic screening, and a lot of people pour the money for the get the information about the genetic screening of the tissues. But this paper published in the Nature Review Cancer in 2015 said that the incomplete biological understanding of the relationship between phenotype and cancer genotype will need a new technology like a cell culturing or the PDX or organoid or other systems. So that's why we want, and also the Samsung Medical Center developed the avatar mouse system, and they acquired the patient glioblastoma and culture the glioblastoma in the human mouse, and then grow them to mimic the phenotype of the patient with the mouse. And then it works pretty well, but as you can easily imagine, it's pretty expensive. It takes a lot of time to grow the mouse, and it's really, really expensive, very difficult to handle. So many people start to thinking about the culture the patient's cells directly in the devices. So we are now also working on it. So many people think about the petri dish or transwell or even the suspended cultures because the patient cancer cells are really, really difficult to handle. And other problem is that when we culture the patient cells in the petri dish, and when the patient cells touch the substrate of the dishes, they dramatically change its phenotype. So some people use the suspended culturing, but it also dramatically decreased the viability of the cells. So we suggested, we built AIM Biotech in Singapore with and we developed the microfluidic culture platform based on the paper published in the Nature Protocols. And it has the microfluidic devices with the extracellular matrix inside, and we culture the cells variously with the three-dimensional structures. And this is one example of the devices, and it has it is the mass producible, absolutely mass producible, and it has the channel for the medium and channel for the ECM, and we usually mix the patient's cancer cells with the ECM and fill this region and culture them in the microfluidic platform. And absolutely that system can be this is Operata high throughput screening system. So we can directly use our platform for the Operata system, and they can measure or they can take images, and this is a 64 array plates to enhance the throughput of our analysis, and this is fabricated devices. So our concept is that acquiring the patient cells, and usually we can acquire the 1 million cells from the patient's tissues, and use the 1.1 million cells for the genetic screening, and we can use the 1.1 million or 0.2 million cells for the drug screening about the 8 or 16 drugs at the same time. And we can directly acquire the very nice information about the patients. And I think I have, 3 minutes left. So this is one example. This patient has the renal cancer, and they has the metastasis in the lung. So he is the medical doctor in the Samsung Medical Center, and Dr. Nam acquired the cells from the kidney and from the lung, and then do the genetic screening. And he found that the genetic screening can differentiate the primary site and the meta site. And you can see the EGFR signaling or SRC signaling. The primary site is a orange color, and the blue color means the metastasis site, and they have different phenotype of the cells. But when we do the direct screening of the cells, when acquire the direct information about the afatinib or dasatinib related to SRC signaling, and afatinib related to EGFR signaling works pretty well for this specific patient. So in this case, we this is our information, so we can acquire that. Okay, afatinib or dasatinib works pretty well, will work pretty well for this patient. But we are sorry for losing this patient because he was too old and we have several other problems. But we cannot make a decision to cure the patient with the dasatinib. But we developed the avatar mouse and cure the mouse with our selection, and finally acquired that this very nice feature to cure the avatar mouse for this specific patient. So this is current status of my research, and we have also another interesting devices, but I do not have more time. So let me briefly summarize it. And this is my company is this work is related to the company, and I have plenty helpers and collaborators, and also they are my students. Thank you very much for listening.
Thank you much for showing us very advanced cell counting and translation system based on the microfluidic device. Okay, now floor for questions. Do you have any questions or comments? Please explain more about the external diagnostic.
Excellent.
Yeah, if you are not ready to disclose, then you can just share with the concept, only concept.
Sorry for that. I have some, but I do not seriously think about the exosome right now because the Professor Tung-Soo is more better for the exosomes. And then I have one news from the company just built. Several companies are built to commercialize the exosome related to some diagnostic devices. But honestly speaking, I do not seriously think about the exosome right now. So I'm more focused on the CTC, not the exosome. Sorry about that.
Okay, do you have any questions or? Okay.
Thank you very much.
Professor Kun from Commercialization of Nanointerstice. This session is going to discuss on the laser pressure, function, and single
Okay. So thank you for invitation for this summer school. And today I'd like to talk about new single cell isolation method we have developed in our lab. So basically we want to use a laser to isolate the single cells from the pathology slide. So let me talk about the story. So I think most of you know that the genomics has been progressed fantastically